Hello, everyone. My name is Doug Chapnick, and I'm the founder and CEO of a biotech company in Colorado called BioLoomics. And I'm here today to tell you a little bit about a project we just started in collaboration with the KIF1A organization to apply our protein evolution in a microscope technology to advance KAND therapies. So our project has three parts. In the first part, we aim to build an assay, a tool that we can search for drugs that could be KAND therapies with. And then once we have that assay, we can transition to another approach we're after, where we search for novel targets that could be drugged, that we might potentially already have drugs or have FDA approved drugs that can be applied in the KAND space. And ultimately, our goal is within 6 to 12 months to find a drug that is suitable to test in animals. And if it's successful there, we can enter into clinical trials. Our company is relatively young. We started in 2019, and since then we've had 2 rounds of debt financing, and we're headed into our Series A this year. We only have 6 members, and in all that time, we've been working on inventing this technology, which is called evolution in a microscope, to apply to build not just drugs, but drug discovery assays as well. And our story started in a Department of Defense project that was funded at University of Colorado that I participated in and another team member participated in, called Rapid Threat Assessment. And in this project, we were given $14 million to come up with a way and a technology that's capable of finding antidotes for novel bioterrorism threats that's independent of the literature, so no mechanism of action. And after the 6 years of that project, which ended in 2018, we concluded that mechanisms are actually not absolutely required to find antidotes. And what is required is absolutely massive parallel testing of candidate drugs that could be antidotes. And so when the project ended, we formed BioLoomics. And BioLoomics, rather than having this Department of Defense angle where we're looking for antidotes, we've transitioned to apply the lessons learned in the new technology we've built to do massively parallel testing for therapeutics in the therapeutic space. When we search for our assays and drugs in our gene libraries that we create, and if you want to chime in and learn a little bit more about the details of the technology, please attend my scientific talk that I'm also giving at this KAND conference, where I go over our approach, how our technology works, and how we're applying it to meet our goals. In a nutshell, what we do is we use human cells in petri dishes to test all different types of random designs of how to build a tool or how to build a drug. So using imaging, we can measure in parallel all the different behaviors of all these different designs, millions to billions at a time. And what we can weed through are designs that don't work the way we want versus the rare designs that do work the way we want. So if you step back, what we're doing is we're failing millions of times to succeed only a handful. But we're doing that all at once rather than in series, which is what traditional R&D is doing. And another way that we're non-conventional in drug discovery is that we don't rely on a detailed mechanism of action for a particular disease. Whereas conventional drug discovery spends about 5 to 10 years determining a mechanism of a disease, and that's usually done in academia, and then a transition is made to build the drug screening tool and then to ultimately screen drugs and find a candidate drug for a particular disease. And so that whole process takes about 15 years. And our way is a whole lot faster. By deleting this whole R&D step where we don't try to construct a detailed mechanism, we're able to jump right into searching for the drug screening tool. And then in 3 years or less, we're able to find a drug that can enter clinical trials. And to give you an example of how you can leverage less information, I put this sort of real world thing that we all kind of went through recently. So if you looked at Tokyo in 2019 at a street corner versus Tokyo in 2020, you would have found lots of people in 2019 and basically no people in 2020. And we know, stepping back, that the cause of this, of the change in the health of the city from these street cameras, was the virus. But just using the street camera alone and the pictures it takes, we can determine when Tokyo is abnormal versus normal. And much in the same way, we take pictures of cells to determine cells are abnormal or normal. And so our strategy for finding an assay for KIF1A mutants that we can then exploit to search for drugs is to take pictures of all different biosensor designs that measure all different biochemistry in cells, introduce our KIF1A mutation, look for the cells that change, and then in our second phase, we're looking for a drug that reverses that. So although we don't necessarily know how our tools work in a detailed way, and we don't know how KIF1A malfunctions cells in a detailed way, we can identify these states using pictures of cells. And so this is how we can construct our search for both biosensor assays, so assays for drug discovery, and also ultimately for our goal to find drugs. And our tech is well equipped to make an impact specifically for KAND, because KAND has relatively few to no assays. It has a very limited mechanism of understanding, aside from KIF1A mutations drive the phenotype of the disease. And there are no drug targets beyond KIF1A that we're aware of. And so our tech can build tools, it can does not require the mechanistic understanding, and has the potential to identify drug targets. So it's well matched to the features of KAND. And so that's basically my brief talk about our project and how we just kicked it off about 2 weeks ago. And how you can help is you could donate to kif1a.org to help us grow the team for the project. And if you're in the crowd and you have access to grant resources or know of grants that we might be a good fit for, help us fund this project with grant money. Our ultimate goal, once we develop our tools, is to apply for an SBIR grant through the NIH NCATS department. And so if you have any ideas that can help us in our grant resource goals, you can contact me at Chapnick@bioloomics.com. Another way you can help us is by following us and spreading the word. So share our mission on social media. We're on LinkedIn, Facebook, and Instagram. And I also want to extend a special thanks to the organizers of the KAND conference and to the KIF1A organization for inviting me to represent BioLoomics and tell you about our project today. With that, I thank everyone for listening, and I'll take questions. Thank you.